Yeah. So, Kirsten, we are very pleased to having you here, and also for your accessibility to answer some questions for the wide public about your work. So, could you make a small resume of the work? For example, you were one of the very important persons in the Artemisinin project. Could you summarize a little bit what this has meant to you, how this is the importance of this work for health, etc.? So,
yes. The work was started in Jay Kiesling's lab. The intent was to take the basic research that they were doing in the laboratory setting in the university and bring it to use it to create a real product that could make the world better. And they noted that one of the most useful and powerful isoprenoid products to harvest the fruits of the isoprenoid pathway manipulation they had done was to make the drug artemisinin, which is a powerful anti-malarial drug. It's effective in most cases against malaria, much more effective than the old line drugs to which there's extensive resistance. The problem at the time in the early 2000s with artemisinin was that the supply was limited and fluctuated dramatically from year to year. And as the supply fluctuated dramatically, so too did the price. And that made this powerful anti-malarial drug out of reach of the people who needed it most for cost and supply reasons. And the source of artemisinin, the only source of artemisinin, was extraction from a plant called Artemisia annua. And as I said, the supply was too scarce and fluctuating from year to year. So their thought at the time they initiated this work was to genetically engineer yeast. Initially they started with bacteria, but they quickly moved to yeast. Genetically engineer yeast, the same yeast that's used to make bread and beer and wine, to make instead artemisinic acid, which could be readily converted into artemisinin. And the work was started in a university laboratory, but in 2005 it was spun out into a new company called Amyris. And over the course of the next 3 years, through the end of 2008, we did extensive genetic engineering in the company to produce to generate yeast strains that were capable of making vast quantities of artemisinic acid. By the end of 2008 or early 2009, the strain was sufficiently good for manufacturing and scale-up, and at that time we transferred the yeast strain to Sanofi Aventis, who developed the fermentation scale-up technology and the large-scale chemistry needed to make vast quantities of artemisinic acid in large stainless steel vats and then to convert it into artemisinin. Starting this year, it's now being manufactured by Sanofi Aventis, tableted and packaged as an artemisinin combination therapy. By the end of the year, we expect to make 70 million cures for malaria that will be distributed in the developing world for those who need it most.
In this work, as you said, it was very important the collaboration between a university setting, an academic setting, and the industrial setting. How do you see this collaboration? This could be very interesting because in this country, not many industries are, well, we don't have many industries, but not many of them are looking to academic research. So could you explain a little bit the benefits of this collaboration, both for university and for the academic, because I think there is a kind of symbiosis in this research. Could you, this, I think it would illuminate many persons in this country.
Absolutely. In order for to develop a thriving industrial fermentation corporate community or industry, it's absolutely necessary to have a close interaction with academia. And the kinds of research that can and do go on in companies is really quite different from the kind of research that can and does go on in academia. But there's a very nice interplay between the two, and they each can help out the other. We found that with our company the way that it came out of a university lab, but we continue to benefit from university labs as collaborators and consultants, and by looking in the literature at the developments that are taking place in academic labs, which are much more exploratory and long-term. The work is much more fundamental and foundational, but we couldn't possibly do what we do in industry without the knowledge base that's generated by academic.
So that in spite of this industrial participation, the sponsoring and the funding of basic research in an academic setting continues to be very important.
Absolutely, yes.
Okay. So thank you very much. I think it was all very nice from you to answer these questions.
Thank you very much.